I'm going to explain the structure of the pituitary gland, the histological structure. The pituitary gland is a major gland in the endocrine system. It's often referred to as being the master gland. And what I'd like you to understand at the end of this lecture is firstly what a hormone is, or a chemical messenger is, and then also understand that the pituitary gland is really two different glands because it's derived from very different embryological sources. It's important also to understand that the pituitary is connected to the hypothalamus, both by a very special portal blood system and also by having neurons extending from the hypothalamus into the pituitary. And these neurons are forming a very important part of the pituitary gland, or at least one part of the pituitary gland. And then finally, within the pituitary, there are a number of different cell types. It's important that you understand different cell types that are there and what their secretory products are. At the end of all studies of endocrine systems, then they are endocrine glands. They consist of endocrine glands, endocrine cells that have a variety of different functions. They're derived from a variety of different places in the body during embryological development. They secrete their product into the bloodstream, and they have hormones that regulate the activities of many organs of the body. They also function along with the nervous system, but their effects are often slower and more prolonged. And their vital function is to maintain homeostasis and control a number of other functions within the body, general functions such as growth, development, and also reproduction. Well, let's first of all look at a hormone, its chemical nature or structure. Hormones are the secretory products of the endocrine cells. Sometimes other cells, not part of the endocrine system, also secrete products that we refer to as being chemical messengers. Hormones can be classed chemically into three types that I'll go through in a moment. But I just want to make sure we know the difference between endocrine secretion, paracrine secretion, and autocrine secretion, and also exocrine secretion. In a separate lecture on exocrine glands, I explained that these glands are derived from epithelial surfaces. The epithelium grows down underneath the epithelial surface within connective tissue, the lamina propria, and there these cells develop into secretory cells, and they maintain their connection to the surface through what becomes the duct of the exocrine gland. In the case of endocrine glands, the cells lose their connection to the surface. The duct disappears. And so these secretory cells then release their products, hormones, into the surrounding interstitium. And thereby, the product is taken from the interstitium into the bloodstream. And so these hormones can travel right throughout the body, and they can affect target organs at some distance from the original source of the hormone. That's a true endocrine gland, true endocrine secretion. Sometimes cells can secrete hormones or products that affect their own function. That's called autocrine, self, being autocrine meaning secretion, secreting to affect oneself. Sometimes these cells can secrete hormones that move along the interstitial fluid and affect neighbouring cells. That's referred to as paracrine secretion. But in this lecture, I'm going to refer mainly to only true endocrine type secretion. Well, hormones are of three types. One, they can be steroid hormones, or they can be peptide, protein or polypeptide hormones. Some hormones can be derived from amino acids. And here on this slide I've listed the various locations that these hormones and the organs are secreting. And I'll refer to these in a later lecture. But the reason why I'm showing you these three different classes is because the chemical nature of these hormones is different. And therefore, sometimes when you see the locations of these cells in the various endocrine glands of the body, you can stain for these different sorts of hormonal products. And these stains can often be used to distinguish the various types of endocrine glands. As we'll see in this lecture on the pituitary gland, we will see that various stains are used because they differentiate the components of the secretory cells. And again, as I said, this will be repeated in another lecture on the other endocrine glands. Well, when a hormone is secreted, it can affect the cell at two sites. One, it can bind to a receptor on the surface of the target cell. In some other cases, it moves into the cell and it reacts with receptors within the cell itself. And the cell biologists and the endocrinologists will describe these processes in other courses. It's important to understand a little bit about the regulation of the endocrine system. We call it a feedback system, a feedback mechanism, whereby the stimulus then goes to the endocrine cells, and the endocrine cells secretes a hormone, and that hormone has an effect. And when the effect then feeds back on the original stimulus and diminishes that stimulus, we call that negative feedback. And that's the most common feedback mechanism we have in the endocrine system. Rarely, though, sometimes we have a positive feedback system, which is the opposite. Well, let's look at the pituitary gland. It's often called the hypophysis. Here is, on the left-hand side, a diagram representing the structure of the pituitary gland, and on the right-hand side is the histological section through the pituitary gland. And it's important that we go through a number of different components. The pituitary gland is really a very small pea-shaped gland. In males, it's about 0.5 gram in weight. In females that have had 2 or more children, it can be about 1.5 grams. So there's a difference in size. The pituitary gland sits in a depression called the salica turcica in the sphenoid bone at the base of the brain, at the base of the skull. And as you can see in both these sections, first the section of the image or histological section through the pituitary gland on the right and the diagram on the left, the pituitary gland is connected to the brain, it's connected to the hypothalamus through a structure we call the infundibulum. And that's a very important connection because the hypothalamus influences the secretory nature and products coming out of the pituitary gland. Well, the other thing you see in this diagram is that the pituitary gland has two different components, two different major components. On the right-hand side, on the histological section, the far left component of that section is a lighter purple colour compared to the rather more enlarged component on the right-hand side of this section. And this coloration defines or distinguishes the two parts of the pituitary gland. The lighter stained area is the posterior part of the pituitary, or the posterior pituitary gland. We call it often the pars nervosa, or the nervous part, or the neurohypothesis, because another name for the pituitary gland is the hypothesis. And on the right-hand side, the darker stained region represents the anterior pituitary gland, the bit towards the anterior of the body. It's often called the adenohypophysis, adeno meaning glandular. It's often called the pars distalis as well. Now, both these components of the pituitary gland are derived from two separate embryological locations. And that's why they should be really considered two separate glands. The pars nervosa, the nervous component, is derived from a downgrowth from the third ventricle of the brain. So it's derived from neuroectoderm. The anterior pituitary is derived from an out pocket of the oropharynx, or Rathne's pouch. So it's derived from oral ectoderm. So these two differences are the reasons why, histologically, they're very different when we look at them using a microscope. Now, the diagram on the left-hand side also shows a number of different structures associated with the pituitary gland. There is part of the pars distalis, or the anterior lobe of the pituitary, that extends up and wraps around the infundibulum, the part of the stalk or the connection to the hypothalamus. There's another part there called the pars intermedia. This is of little relevance to us, so I'm not going to cover it in any detail. Have a look at the hypothalamus, that yellow stained component. In the hypothalamus, you can see two nuclei labelled. These nuclei are called the paraventricular nucleus and also the supraoptic nucleus. They're going to be very, very important structures that I'm going to refer to in the next couple of slides. This diagram, again, appears to be rather complicated, but there's only a couple of very simple stories that I want to emphasise. First of all, have a look in the hypothalamus. Again, look at the paraventricular nucleus and the supraoptic nucleus. A nucleus in the brain is a collection of nerve cell bodies, and they're shown here in this diagram. Notice, though, that the cell bodies project their axons down the infundibulum, or the pituitary stalk, into the location of the posterior pituitary, the neurohypophysis. Now, these neurons are neurosecretory. They don't transmit an impulse. What happens is that oxytocin and ADH, two hormones, are produced by these two nuclei, by the cells in these two nuclei. Oxytocin is produced by mostly the paraventricular nucleus. ADH is produced mostly by the supraoptic nucleus. And those secretory products travel down the axons and are stored in axon terminals in the neurohypophysis. And we'll see that in a later slide. So the secretion of oxytocin and ADH is from neurosecretory cells directly into fenestrated blood capillaries indicated there in the diagram. The blood supply to this region comes from the inferior hypophyseal artery. So that's the major component of the paraventricular nuclei and the supraoptic nuclei providing secretory products into the posterior pituitary. There's also another group of nuclei that you see in the hypothalamus as well. Got a very long name to it. Hypothalamo-hypophysiotropic nucleus, or nuclei. They contain neurons that secrete releasing factors. And those releasing factors get taken up by a capillary bed I'll mention in a moment up in the hypothalamus. And those releasing factors, when they get into the capillary bed, can affect the secretory products from the anterior pituitary. And I'll go through that in a moment. If you look at the adenohypophysis, the anterior pituitary, you notice that in the diagram, there are three cell types represented. Basophils, acidophils, and then cells that don't take up any stain. And this is because in the anterior pituitary, the typical glandular epithelial component of the pituitary gland, there are different cell types based on their staining characteristics. We have cells collectively called chromophils These are ones that have an affinity for colour, and they're the basophils or the acidophils. Then we have the chromophobes, which make up a large proportion of the cells that don't take up the stain. And we're not quite sure whether these have any functional significance or whether or not they happen to be the chromophils that are just going through a process of resynthesizing their secretory products. But I'll show you these cells in more details in a moment. But just imagine now that the anterior pituitary is full of these different secretory cells that secrete different products we'll see in a moment. And those cells secrete into the vascular system. So it's important now that I explain the vascular supply to the pituitary gland. If we go back to the hypothalamus region, you'll see just at the top a vessel called the superior hypophyseal artery. Now this artery branches and it provides the hypothalamus with a network of capillaries. It's called the primary capillary plexus or network. And it's in just the upper part of the infundibulum, that stalk that connects the pituitary gland to the hypothalamus. That's a very important primary plexus of capillaries because those secretory products, the releasing factors I mentioned earlier from those hypothalamo-hypophysiotropic nuclei, are secreted into that primary capillary plexus. From that plexus, the blood then flows down through portal veins along the infundibulum, and then they all supply the anterior pituitary. They don't go to the posterior pituitary. So this is a situation where the releasing factors that are secreted from those nuclei in the hypothalamus, I'm not going to try and say that word again, they pass down through the capillaries and can affect the secretion of the cells in the anterior pituitary. These releasing factors can either stimulate cell secretion from these cells, and they're very specific what cells they actually stimulate, or they can inhibit secretion from these cells. So there is this control of the secretory products coming out of the anterior pituitary by neurons in the hypothalamus. The posterior pituitary, as I said before, does not receive these portal vessels, and therefore the secretion products coming from the neurosecretory cells in the posterior pituitary are unaffected by these releasing factors. This hypothalamic hypophyseal portal system is an extremely important concept to understand, and it's an extremely important piece of histology and microanatomy influencing the function of the anterior pituitary gland. Well, here is a little summary of, first of all, on the right-hand side, the components of the pituitary gland, and on the left-hand side, those components in diagrammatic form. And I think it's important for you to fully understand that there are these components in the hypothalamus, neurons in the hypothalamus, understand where they project their axons and what the function of these projections and their products are in the control of the other glands that we'll see from these secretory products. And understand also the concept of epithelial cells in the anterior pituitary staining differently and influenced via releasing factors coming down that hypophyseal or hypothalamic hypophyseal portal system. Let's have a look at the anterior pituitary in a bit more detail. On the left-hand side are these very pretty cells stained, either acidophils or basophils. And as we'll see in a moment, different cell types are within these two types of stained cells, and they secrete different hormonal products. Those orange-shaped structures you see or coloured structures you see in the section are actually blood vessels, blood capillaries. The pituitary gland, both the anterior pituitary and the posterior pituitary, being an endocrine gland rather, are extremely vascular, as you'd expect, because those blood vessels take away the secretory product. When we look at the posterior pituitary, it's a different sort of arrangement. It's a different sort of histological structure. If you look down the bottom slide, I've labelled a pituocyte and also the herring body. If you look very carefully at the herring body, you can see little tiny brown granules. These represent vesicles containing the secretory products from these neurons, whose cell bodies, remember, are located in the paraventricular nucleus or the supraoptic nucleus up in the hypothalamus. So these herring bodies are actually the terminal ends of these axons, and they swell up as they fill with oxytocin and or ADH. And those secretory products are going to be released into nearby blood vessels, neurosecretion. And those blood vessels are going to be fenestrated. And that's a type of capillary that can easily mop up the secretory products from the interstitium and then carry it to the target organs located somewhere else in the body. The pituocyte is a supporting cell. These axons are not myelinated. The pituocyte is not a Schwann cell or an oligodendrocyte or a cell that's involved in myelination or supporting unmyelinated fibres. It's like an astrocyte, because if you think about it, the nuclei that contain the cell bodies of these axons you see in this section through the posterior pituitary, those cell bodies are protected within the blood-brain barrier. And when the axons then project down through the infundibulum into the posterior pituitary, they then break through that blood-brain barrier. Well, the astrocyte-type cells here, the pituocytes, maintain in some form the integrity of that blood-brain barrier, because what they do is they not only wrap around and support the axons, but they also wrap around the blood capillaries, the fenestrated blood capillaries, and they then create this barrier and protection. So as I said a couple of times, they're just like an astrocyte that you see in the central nervous system. They have a similar role. We can now summarise the structure of the pituitary gland, and then we can start to assign different secretory products from those different components of the pituitary gland. Firstly, I did mention earlier that the posterior pituitary, the neurons in the supraoptic and paraventricular nuclei, synthesize ADH and oxytocin. And they're released in the posterior pituitary into those fenestrated capillaries I've just mentioned. And those two hormones have effects on various target organs that I've listed there, and just basically their general function of those hormones, what they actually bring about in those organs. And this is just a summary. Endocrinologists will deal with these sorts of roles these hormones have in far more detail. Here, I'm only giving the histological background and basis for these hormonal products. If we then look at the acidophils in the anterior pituitary, then we can see that there are, first of all, hormones we group together as being lactotropes, secreting prolactin. Or they could be somatotropes involved with growth hormone secretion, who act via the liver and via somatomedins. And you can see there the target organs for each of these hormones. The lactotropes and the somatotropes, or at least the somatotropes, occupy about 50% of all the cells in the anterior pituitary. The lactotropes, those that secrete prolactin, occupy between 15 and 20% of the cells in the anterior pituitary. The basophils, by far, are the busiest group of epithelial secretory cells in the anterior pituitary. And again, I've listed the general functions of the different hormones secreted by these basophils. The corticotropes that affect the adrenal cortex, they too occupy about 15 to 20% of all the secretory cells in the anterior pituitary. The thyrotropes occupy just a small percentage, perhaps 5%. And the gonadotropes occupy, again, only about 10%. So there is this variation in the numbers of various cell types in the anterior pituitary that secrete all these different hormonal products. Thyroid-stimulating hormone, follicular-stimulating hormone, luteinizing hormone, and adrenocortical-trophic hormone. And I'll refer to these hormones in more detail when I actually cover all these different organs in other lectures. I'd like to now just point out the variation in this particular slide of all the organs affected by the pituitary, by hormones secreted by the pituitary. And I want you to recall what I said earlier, that endocrine cells secrete hormonal products that are carried via the bloodstream to target organs at locations a long distance away. Well, when you look at this diagram and you look at the various organs represented, it makes that point very, very clear and very, very obvious. In summary then, it's important that you understand the structure of the pituitary gland, particularly the two components of the pituitary gland. It's important you understand the different types of secretory components within both the posterior and the anterior pituitary, and the secretory cells within them, and what the secretory products are from each of those cells, and the general target organ and general function of those different hormones. It's also very, very important to understand that hypothalamo-hypophyseal portal system, and that how that allows the communication between releasing factors from the hypothalamus and also factors from other parts of the central nervous system, how those releasing factors can either stimulate or inhibit cells in the anterior pituitary and therefore control their function. So thank you for listening to this lecture. I hope you now have a very good understanding of the pituitary gland and the general features of hormones and the endocrine glands.